Welcome to the class. Today's topic is blood cell count and anemia. Introduction. Hematology is the study of blood and blood smear, one of the most basic and yet most reliable ways of evaluating blood for multiple conditions of disease. The river of life called blood is composite fluid connective tissue, which is diverse in function and the most important tissue in the human beings. This tissue provides every necessary element starting from food to sustenance of living cells, and more importantly, it is a means to get rid of harmful waste materials. The other functions are maintenance of body temperature, defending the cells from invading microbes in case recovering from the attacks. This is mesodermal in origin. It circulates with pumping organ, the heart, all over the body to maintain homeostasis. Different types of blood cells and their roles in human body. Blood comprises of two parts. One is plasma, other one is foam cells. Foam cells are of three types: platelets, RBC and WBC. WBC are two types again, granulocyte and the agranulocyte. Agranulocyte comprises of two types of cells. One is monocyte, other one is lymphocytes. And the granulocytes comprises of three cells: eosinophils, basophils, neutrophils. Lymphocytes: B lymphocyte, T lymphocyte. B lymphocyte produces antibodies, while T lymphocytes comprises of four cells. They are helper T cells, killer T cells, memory T cells, suppressor T cells. Figure 1. Blood and its component in human beings. Highlights are parts of immunity. A blood cell, also called hematopoietic cell, hemocyte, is a cell produced through hematopoiesis from bone marrow cells of skeleton system. Figure 2. Hematopoiesis in different parts of human body. Figure 3. Hematopoiesis of different cells in bone marrow cells of human body. Usually blood cells count is part of a routine checkup or screening or as a follow-up test to monitor certain treatments. It can also be done as a part of evaluation based on patient symptoms. Hence, blood count is much before diagnosis a disease, particularly related to the metabolism malfunctions. For example, in high WBC count called leukocytosis may signify an infection somewhere in the body, or less commonly it may signify an underlying malignancy. A low WBC count called leukopenia may point toward a bone marrow problem or related to some medication such as chemotherapy. A doctor may order the test to follow the WBC count in order to monitor the response to a treatment for an infection. The components in the differential of the WBC count also have specific functions, and if altered, they may provide clues for particular conditions. A low red blood cell count or low hemoglobin may suggest anemia, which can have many causes. Possible causes of high red blood cell count or hemoglobin erythrocytosis may include bone marrow disease or low blood oxygen levels, hypoxia. A low platelet count called thrombocytopenia may be caused of prolonged bleeding or other medical condition that affect the production of platelets in the bone marrow. Conversely, a high platelet count called thrombocytosis may point toward a bone marrow problem or severe inflammation. Total and differential counting of leukocytes and the RBC counting. The cost-effective and common method of blood counting is by using hemocytometer. Figure number 4. Hemocytometer. 1. Different components. 2. Diagrammatic representation and its part. Third, part used to count RBC and WBC. Pictures of the blood cells included. And fourth, general norms for counting and excluding during counting of the cells in the blood. Blood was prepared by diluting with citrate 4% weight by volume of sodium citrate at a specific ratio for each type of cells to be counted. Then the solution was introduced into the counting chamber of the hemocytometer, figure number 4. Cells count can be done at different chambers. The height of the chamber is 0.1 mm. Then total length of one side of 5 RBC chambers, that is 4 corners and 1 center. RBC snap is given is 1 mm. Therefore, RBC count, that is cells by microliter. RBC count, that is cells per microliter of blood, equal to mean RBC count for 5 boxes into 25 divided by volume of 5 into 5 RBC region in microliter multiplied by dilution factor, that is 1:200. WBC count cells per microliter equal to mean WBC count for 4 boxes divided by volume of 4 WBC region in microliter bracket 0.4 into dilution factor, that is 1:20. According to the American Association for Clinical Chemistry, AACC, the test is almost always a part of complete blood count CBC test. A CBC test measures the number of all components in the blood, including red blood cells, that is RBC, white blood cells WBC, haemoglobin, haematocrit, platelets. The haematocrit is the volume of red blood cells in the body. A haematocrit test measures the ratio of RBCs in blood. The haematocrit measures the fraction of the blood that is made up of RBCs. It reflects the combination of total number of RBCs and the volume that they occupy. Normal ranges in male it is 40 to 52%, in female it is 35 to 47%. One of the changes seen in the pregnancy is drop in haematocrit. This occurs because although the production of RBC does not change greatly, the plasma volume increases, that is, RBCs are diluted. Alternatively, a low haematocrit can reflect a drop in RBC production by bone marrow. This may be attributable to bone marrow disease, that is, damage by toxins or cancer, or due to decrease in erythropoietin, a hormone secreted by kidney that stimulates RBC production. Decreased RBCs may also be result of reduced lifespan of RBCs, that is, chronic bleeding. A high hematocrit value may truly reflect an increase in the fraction of RBCs, that is, increased erythropoietin attributable to a tumor of RBC called polycythemia rubra vera, or it may reflect a drop in the plasma component of the blood. For example, fluid loss in burn victims. Platelets are small cells that circulate in the blood and form blood clots and allow wounds to heal and prevent excessive bleeding. Table number 1, the complete blood count. Blood components abbreviation used, reference range. White blood cells WBC 4500 to 11,000 per mm cube. Red blood cells RBC male 4.3 to 5.9 million per millimeter cube. Female 3.5 to 5.5 million millimeter cube. Hemoglobin HGP male 13.5 to 17.5 gram per deciliter. Female 12 to 16 gram per deciliter. Hematocrit HT male 41 to 53%, female 36% to 46%. Mean corpuscular volume MCV 80 to 100 micrometer cube. Mean corpuscular hemoglobin MCH 25.4 to 34.6 pg per cell. Mean corpuscular hemoglobin concentration MCHC 31% to 36% Hb per cell. Platelets 1,50,000 to 4 lakh per millimeter cube. Total leukocyte count TLC is the total number of all the WBCs in the blood. Differential leukocyte count DLC. White blood count is comprised of several different types that are differentiated or distinguished based on their size and shape. The cells in a differential count are granulocyte, lymphocyte, monocyte, eosinophils and basophils. Differential WBC count. For differential count of WBC, blood smear stain with GEMSA is required. WBC differential count has been widely accepted and used by clinicians and is generally considered to yield clinically useful information in health and disease. Amount of each WBC type in blood. Norman range table 2. The Norman range of WBC cells. Percentage of WBC absolute in billion per liter. Neutrophils 45 to 75, 2 to 8. Lymphocyte 20 to 50, 1 to 4. Monocyte 1 to 8, 0.1 to 0.8. Eosinophil 0 to 6, 0 to 0.5. Basophils 0 to 1, 0 to 0.3. Figure 5. Different types of WBC emphasized for differential WBC counting from the blood smear. RBC count and anemia. RBC count is generally meant to know the number of RBCs at a particular period of an individual. The test is important because RBCs contain hemoglobin, which carries oxygen to your body's tissues. The number of RBCs can affect how much oxygen tissues receive. The tissues need oxygen to function. Symptoms of an abnormal count. If your RBC count is too high or too low, you could experience symptoms and complications. If low RBC count, symptoms could include fatigue, shortness of breath, dizziness, weakness or lightheadedness, particularly when the person changes position quickly. Increased heart rate, headaches, pale skin. If a person have a high RBC count, the person experience symptoms such as fatigue, shortness of breath, joint pain, tenderness in the palms of the hands or soles of the feet, itching skin, particularly after a shower or a bath, sleep disturbances. Anemia is a condition in which there are not enough healthy red blood cells to carry oxygen throughout the body. Conclusion. The blood is the most important fluid connective tissue. This tissue as a reference point, many abnormalities in the body could be thoroughly understood. Many chronic or congenital diseases could be easily screened out and adopt appropriate medications to relieve from the suffering by a medical practitioner. But screening should be done in a qualified and well-equipped laboratory and thoroughly examined by an expert on the matter. There in the clinical laboratory, blood will be collected from the individual and the clinical staff will give the reports of the test. but in the educational institutes, the study of blood called hematology could be done with the help of hemocytometer, which is used for counting both RBCs and WBCs. The blood smear is used for differential counting and change in the RBC structures to know the cause of anemia. Hence, the theory behind the hematology and the screening of the blood is to be thoroughly understood, because this will help us to know the abnormalities and give suggestions on the methods to ourselves and anybody in the future.